Hi everyone, I'm a yeast. My name is Saccharomyces cerevisiae. I have a twin who is very sad because he doesn't have the methylmethane sulfonate sensitivity gene. So his name is MMS2 mutant. We're here because scientists are very curious of what happens when both of us are exposed to a drug called fluconazole. So, what is MMS2? MMS is a subunit of ubiquitin conjugating enzyme complex. It binds to ubiquitin and present into substrate, contributing to ubiquitin protein transferase activity. It is thought to be involved in error-free post-replication repair. Fluconazole is an antifungal drug that inhibits cytochrome alpha methylase enzyme. This prevents conversion of linosterol to ergosterol. In the fungus, this effect is thought to interfere with reuptake of purine bases into the cell. So we hypothesize that if purine bases can't enter the cell, it stresses DNA replication. But don't worry, our superhero MMS2 comes in to help here so that DNA is replicated correctly. As DNA replication stress got higher, more MMS2 are produced and intracellular level of MMS2 goes up. So, in the experiment, scientists exposed us to the cell wall or cell membrane stresses in different concentrations. No stress control is positive control. Plates with different concentrations of fluconazole and caffeine were tested. Wild type and mutant strains were prepared in 10-fold dilutions and spotted onto plates. Then we were incubated at 30 degrees Celsius. Results were taken at 3 days and 7 days respectively. So, what are the results? Positive control plates showed that both strains grow equally and there could be minor growth difference due to variation in pipetting. When exposed to 8 microgram per ml FLC, mutant strain grows less than wild type. The same trend together with reduction in growth for both strains is observed at higher concentrations of FLC. Mutant strain also grow less when stressed with caffeine. This effect is more exaggerated when both stresses are combined. In the experiment, we found that mutant strain grows less than wild type in the presence of FLC. This confirmed that FLC stresses the fungal cell and inhibits uptake of purine bases. Therefore, MMS2 mutants could not cope with DNA replication stress and most did not survive. It was found from previous research that caffeine stresses the fungal cell wall. To restore cell wall integrity, MAPK pathway is activated to produce transcription factors. These start transcription machinery and produces protein that restores cell wall. But it was found that MMS2 is an important factor in the pathway. So, in my mutant twin, MMS2 is absent, so MAPK pathway could not function well and they did not cope well with cell wall stress. So, to sum up, it is not conclusive that the FLC stresses fungal cell membrane, but it stresses the cell wall. Further research could be conducted on other cell wall or membrane stresses and with higher range of concentrations. Thank you for watching.